want to cover is something called, membrane or input resistance. And this is a measure of how many channels are open in the neuron at rest. And it's really a measure of how leaky a neuron is. So if we take the example of high membrane resistance, this is where we have a neuron that has most of its channels closed. And so if you have an incoming stimulation or incoming input, from a synapse, that charge, because those channels are closed, can stay in the neuron and charge the neuronal membrane and depolarize it, and then maybe lead to an action potential. And so the opposite would be an example of a neuron with low membrane resistance. And this is where you have channels which are mostly open. And so now if you have a current come in from say, synaptic input, that charge can then essentially just leak straight out of the neuron, and we're not able to charge the neuronal membrane to generate an action potential. And so using these properties, we can look at changes to intrinsic plasticity. And the way that we do this is through a technique called patch clamp electrophysiology, which is mostly done in brain slices. And so we can cut brain slices from the rodent brain. They're about 300 microns thick. And this then allows us direct access to the neurons across multiple layers. And we can insert glass electrodes, shown here in the arrows, into the neurons to actually record their properties. And because even though we're slicing the brain, in that 300 micron slice, the local circuitry remains well preserved. So we can look at connections between the different neurons in that slice. And so here's an example of where we're taking the brain slice. You can put it under a microscope, and they look like grapes, really, kind of like these grape-like structures under the microscope. And then we can insert our glass electrodes into them. And using this technique, we can actually record their passive properties. So what their properties look like when they're at rest, such as resting membrane potential. But we can also look at their properties when they start to spike or generate action potentials. And because we have direct access to these neurons, we can actually record their input and output relationship directly by injecting current into them. And so we don't actually need synaptic input to record this. And so today I'm going to cover some of the studies which have used this technique to study the effect of rTMS. But before we get there, I just want to remind you that the mammalian motor cortex, whether that's a mouse, rat, or a human, is a layered structure. And so the most simple motor cortex circuit consists of layer 5 pyramidal neurons, which I'm sure you're all familiar with. This is probably the most well-known neuron from the motor cortex. It forms the cortical spinal tract. It also sends connections to other parts of the brain, such as the striatum or the other hemisphere. We also have in layer 2-3 our pyramidal neurons, and these are the neurons which we believe are responsible for the I waves in the MEP. And we also have inhibitory interneurons, which sit both layer 2-3 and layer 5, which provide inhibition onto our pyramidal neurons. And so that's where I want to start today's data, like what happens to intrinsic plasticity in this cell type. So the first study I want to cover, which is still ongoing, is some work done by Carly, who's going to talk about her glia work next. But basically, Carly took the mouse TMS coils I built to stimulate the motor cortex with ITBS daily for 14 days. And then she measured changes to intrinsic plasticity 24 hours post-stimulation relative to control. And so when she looked at layer 2-3 inhibitory interneurons, what she found was that if you look at the membrane resistance or how leaky these neurons were, the brains that were treated with ITBS had a lower membrane resistance. These cells were leaky. So essentially they can't hold charge as well. And so knowing this, we would expect that if ITBS cells which have been stimulated with ITBS have leaky membranes, you would then need a greater current injected into them to generate action potentials. And this is exactly what she found, that you needed more current into these neurons to generate action potentials. And when she looked at the input-output curve, so increasing the amount of current she injected into the cells, not only did these cells require more current to generate action potentials, but when they generated trains of action potentials, they generated a less number of action potentials. So all these changes put together would indicate that ITBS is decreasing neural excitability by changing these properties. If we look at a different example, this is an example done in Germany by Klaus Funke's group. And this study used a small butterfly commercial TMS coil to stimulate the whole brain, but they took slices from the somatosensory cortex, and they were looking at changes 2 hours post-stimulation. And another interesting thing about this study was that they were at changes to different ages. So they looked at juvenile mice and also young adult mice. Sorry, this was in rats. And so when they look at their changes in intrinsic plasticity, interestingly, what they found was that after ITBS is that the resting membrane potential of these layer 2-3 inhibitory interneurons was more depolarized. It's about 16 millivolt more depolarized, and this only occurred in rats of a specific age. When they looked at these cells which had a lower or more depolarized resting membrane potential, they also found that if you looked at the input-output recruitment curve and injecting current into them, that these cells not only had a depolarized resting membrane potential, but they actually generated more action potentials in response to stimulation. And so taken together, this suggests that, at least in this paradigm, that ITBS was increasing neural excitability by changing these membrane properties. If we now look at our layer 2-3 pyramidal interneurons, the cells which we think generate the I wave, we go back to the work that Carly has done, again, where she gave ITBS for 14 days and then looked at the day after the last stimulation. And what she found was similar to the changes found with Klaus Funke, is that in the pyramidal interneurons, that the cells which had been stimulated with ITBS, that these cells had a more depolarized resting membrane potential, about 6.5 millivolts more depolarized compared to controls. And so taken together, this suggests that, at least in this paradigm, ITBS is altering neural excitability by changing the resting membrane potential. And lastly, if we look at our layer 5 pyramidal neurons, the one we're most familiar with, this is some work that I did a couple of years ago, but I took a slightly different approach. So because the coils I made for the rodents are actually quite small, they're only 8 millimeters in diameter, we can actually put these coils underneath the microscope that we're using to record intrinsic plasticity. And so this allows us to actually record the changes to the neurons, both before, during, and after stimulation. And we were tracking changes for the first 20 minutes post-stimulation. And using this approach, what we found was that if we looked at these pyramidal neurons, that they actually had a lower action potential threshold. So the action potential threshold was closer to resting membrane potential. And when we got the cells to fire, we found that on average, the cells would actually have an increased number of action potentials, anywhere between 2 to 5 more action potentials in the train compared to sham. And so when we took this together, this suggested to us that immediately after stimulation, ITBS was able to increase neural excitability by changing these membrane properties. And so this is just a table to summarize the work that's been done in this space. I'm not going to go through it all, but I think what it quite clearly demonstrates is that if you look across the neural network in the motor cortex, you'll find that the rTMS is changing intrinsic plasticity to multiple cell types, and it's changing them at different times post-stimulation, although it is important to realize that the stimulation protocol is not the same between the studies. But nevertheless, if we think that rTMS is changing neuronal excitability, as you would expect, you'd be surprised to see that these changes are occurring on properties that we had never thought of before. So changes to resting membrane potential, which is usually only ever in discussion when we talk about TACS or TDCS. We see changes to, say, action potential threshold. And these are all very powerful regulators of neural excitability, which are completely outside of the synapse. And so what does this mean, and why should you care? Well, like over the last 30 years, we've always thought that rTMS to the brain drives synaptic plasticity. And certainly over the last 10 years, if we look at the animal work, that's providing strong evidence for that. We see changes in synaptic strengths, we see changes in synapse structure. But hopefully what I've been able to convince you today is that it's also driving changes in intrinsic plasticity, neuronal membrane properties, which are regulating action potential output. And so by changing these properties, both passive and active membrane properties, we're actually affecting the number of action potentials that our neurons are outputting. But not only that, we're changing the timing that these action potentials are arriving at the synapses. And so this is very interesting. And my current working hypothesis is that while rTMS can actually drive synaptic plasticity in its own right and intrinsic plasticity in its own right, that perhaps by driving changes in intrinsic plasticity, this is perhaps feeding into synaptic plasticity, because the number of action potentials and the timing that these action potentials arrive at the pre and post synapse are the code or the information that synapses use to generate plasticity. And so it could be that in addition, that the intrinsic plasticity is potentially regulating things like spike timing dependent plasticity, but also changing things like synchronicity and coherence between different brain regions, because if we're changing the number and timing that signals are projecting to different brain regions, we could be having a potent effect on these outcomes. And so where to next? Work from my group is trying to explore and test more protocols, because most of the work previously done has only explored ITBS. We're very interested in looking at changes between young and aged brains, but also between genders, to try and look at how variability might exist across the network to the same protocol. You saw some work from my PhD student, Emily King, where we're looking at trying to understand how maybe changes in rTMS-induced intrinsic plasticity may be regulating synaptic plasticity and metaplasticity. And this is work that's been done with Cliff Abraham at Otago. And lastly, something I think is really exciting, and we've upgraded our equipment to do this, is that obviously we can continue to exploit the really powerful techniques that we have in animals, but we do want to test this in human tissue. And so something that we're interested in doing is taking human tissue. This is an example of a brain slice taken from Andreas Blatsch's group from one of his non-TMS studies. But basically, you can take human tissue that's been taken from patients that have had tissue resected for, say, epilepsy or cancer biopsy. And we hope to take this tissue and put it on our microscope and put the TMS coil underneath it, actually record changes to both intrinsic and synaptic plasticity after stimulation. And so for any ARC DECRA reviewers out there, hopefully you've liked what you've seen today, and when this crosses your desk, you'll review it favorably. And so lastly, I just want to give a shameless plug for a special research topic that I'm running with Kate Hoy and Paul Crokin. If you're doing any work on non-invasive brain stimulation in developing and aging brains, please consider submitting to us, and you can submit through any 3 of these Frontiers journals. So lastly, I just want to thank those who are involved, specifically the funding bodies, which have allowed me to kind of build my own research team to explore these questions of how rTMS might be affecting both synaptic and intrinsic plasticity. Thank you.
Thank you very much, Alex. That was a really interesting talk, and getting down to those basic membrane properties is fascinating. I'm looking at questions here, and we have one from Nigel Rogash. Thanks, Nigel. Great talk. Thanks, Alex. If I understand correctly, ITBS decreased excitability of inhibitory neurons, but increased excitability of excitatory neurons through different mechanisms. Any ideas on how the same pulse could differentially affect different cell types?
Yeah, that's a really good question, Nigel. And I guess the fact is that we don't really know yet. I mean, the properties that it changed, yeah, I think both saw changes in, so in Carly's study, she saw a change in resting memory potential for the pyramidal neurons, but then changes to input resistance and spike firing in inhibitory interneurons, and these are governed by different ion channels. And so, yeah, we don't really know how it might be affecting the different cells. That's something, I guess, that we do want to look at, but I'm not really able to say how it does it yet.
Okay, thanks, Alex. We have time for one quick question, if anyone has one. If not, I might ask a related question. Alex, you mentioned the differences in age that were detected by the Funke group, and I always thought that was really fascinating, because the differences that they found were just around the critical period of development. Would you perhaps comment on what you think is going on to make those changes happen?
So for that one, the speculated mechanism is that around that time, the perineuronal nets are highly plastic, which are, I guess, feeding onto the inhibition onto those cells. I think the working hypothesis from that group is that during that time, rTMS is able to change the calcium binding proteins to a greater extent than during adulthood, although it's not really known why. And so by changing the calcium buffering at that time, which is more plastic, you can get a greater effect on inhibition.